This is the review session for the last third part of the course, which covered the enzymes and metabolism. And we're going to revisit the critical points that will help you to prepare for your final exam. First, we talked about the enzymes, which are the biological catalysts in the cellular systems. And we viewed those enzymes in a perspective of structural biology and kinetics to better understand their mechanisms in the reactions. We've discussed the energy changes in the catalyzed or uncatalyzed reactions and also talked about the transition state using the state function delta G double dagger, which is the free energy of activation. We talked about the Arrhenius equation, which estimates the reaction rate V, which is also dependent on the substrate concentration. And we also talked about the delta G double dagger, which is a critical factor to explain the catalytic reaction. By using Arrhenius equations for uncatalyzed and catalyzed reactions and getting the ratio of that, we could get the enhancement upon the catalysis for that reaction. Next, we moved on to the biological catalyst enzymes, and we also talked about how enzymes can catalyze the biochemical reactions and also their properties that they're using to interact with the substrate. And then we also talked about two different models to explain these enzymatic reactions, which are lock and key hypothesis and induced fit hypothesis. As an example of the enzymes, we talked about the serine proteases in detail, and you should know about their subtypes and the examples of the serine proteases that we talked about in terms of their enzymatic reactions and their substrates and so on. Specifically, we've talked about chymotrypsin, trypsin, and elastase, and what their substrates are and how they are different in terms of their enzymatic reactions and mechanisms. Serine proteases have especially highly conserved and critical residues, which are histidine, serine, and aspartic acids, which form the catalytic triad. And we talked about how they work in the active site and how they are conserved through the serine proteases in terms of their reactions. We also talked about the different kinds of cofactors, such as metal ions, coenzymes, as well as prosthetic groups. In the second part of the enzymatic catalysis, we focused on the different types of catalysis. And these are the 5 different types of those catalysis: acid-base catalysis, covalent catalysis, metal ion catalysis, proximity and orientation effect, and preferential binding of transition site. And you also should know and understand their mechanisms and their differences too. And you should be able to tell which type of catalysis was used for a particular reaction. To better understand the mechanism of catalysis, we revisited the thermodynamics, so you should review thermodynamics and their state functions, as well as their implications in the explaining the catalytic mechanisms. We also talked about the kinetics of the catalyzed biochemical reactions, which can have multiple intermediates through this reaction to get the products from the reactants. And one of them can be the rate determining step or rate limiting step. And also we talked about the rate or the velocity of the reaction or the each steps too, to better understand how the kinetics work and the regulated for such reactions. And you should understand the rate law and the rate constant and how they're important in terms of talking about the kinetics of the enzymatic reactions. And for enzyme catalyzed reaction, there are at least 3 different states where enzyme is free, or there is a complex for the enzyme and substrate or the products. And for between each state, there are rate constants that can define the kinetics of those states. And also from the substrate saturation curve, you can see the relationship between the substrate concentration and the rate of the reaction. And there is a Michaelis-Menten theory, which is really important for understanding and explaining the kinetics of enzymatic reactions. Again, this is so important that you should understand what this Michaelis-Menten theory is and what you can learn out of this theory to better understand the mechanisms of enzymatic reactions. And of course, there's a simplification and assumption that make this Michaelis-Menten theory work, so you should know them too. And later, Briggs and Haldane improved this Michaelis-Menten theory by adding an idea of a steady state. So that's the assumption where the ES concentration is at the steady state. Again, you should understand what they're suggesting and what it means in terms of better explaining the enzymatic reactions, and that's its kinetics. And in part 3 of enzymatic catalysis, we kept discussing the enzyme kinetics and also talk about the different types and the mechanisms of inhibitions. Here you must understand these three important quantities: Km, the Michaelis constant, Kcat, as well as Vmax. And again, the substrate saturation curve can show the relationship between the substrate concentration and reaction rate. And especially when substrate concentration is at Km, the reaction rate will be the half maximal. And you can get the reciprocal from the Michaelis-Menten equation, basically what it is plotted on the substrate saturation curve, to get the Lineweaver-Burk plot or double reciprocal plot. And you must understand how this transformation works and what those x-axis, y-axis intercepts, as well as the slope mean, and how you can how it is beneficial to get such transformation. Then we move to the different types of the inhibition for the enzymatic reactions. We talked about irreversible and reversible inhibitions, and especially focusing on the reversible ones, and also talked about these three different types, which are competitive, uncompetitive, and non-competitive inhibitions. And then we talked about the effect of inhibitors on the substrate saturation curve and how they can affect differently on it depending on their types. Again, Lineweaver-Burk plot is really useful in a way that you can visually see and better understand how different types of inhibition affect the different aspect of the kinetics. And then we move to the metabolism, and especially in part 1, we revisited the thermodynamics to better understand the energetics of the metabolism through its pathway. First, we talked about the definition of the metabolism and their different components. And then we brought back the thermodynamics here to better understand the energetics and the energetic balance of the any kinds of biochemical reactions through the metabolic pathway. And then we talked about the regulations on enzymes by changing its availability or its activity, and there are several different ways to regulate such enzymes activity, such as using the allosteric or the some kind of covalent or non-covalent modifications on the those enzymes, as well as the change of the localization. Then we talked about what oxidation is, as well as the different states of the oxidated carbon biomolecules. And we also discussed the really critical concept here, the oxidation reduction or the redox reactions. And there are basically two half reactions here, one for the oxidation and the other for the reduction. And you must understand what oxidation is and what reduction is, and what are which one is working for the donor or the acceptor for the electrons. From part 2 of metabolism, we focused on the actual metabolic pathways and their intermediates, the substrates, as well as the enzymes that are involved in such pathway. There were 3 components for the central metabolic pathway: glycolysis, TCA cycle or cyclic acid cycle, as well as electron transport chain. And we discussed all these components. So you must understand what these are and what are the players of such components. We also studied the high-energy phosphate compounds, which have a critical role in the metabolic pathway. And we discussed in detail the glycolysis, each step from 1 to 10. And of course, these are divided by 2 stages. From 1 to 5 belongs to stage 1, which is the investment stage. And step 6 to step 10 is in the stage 2, which is the payoff. And again, you should understand each steps, what the intermediates are involved there, and also the enzymes that are making such changes in intermediates. And now the part 3 was about the step after the glycolysis to further make the energies for the cellular system. After glycolysis, there's a pyruvate that are generated, and it can go to largely 3 different pathways. The first one is a TCA cycle, again, or the citric acid cycle, or the lactic fermentation or alcoholic fermentation. And of course, you should know what TCA cycle is and what it does actually in terms of energy generation. And also that happens in the mitochondria, and we extensively talked about the mitochondria, their properties, their structures, and so on. So you should also memorize those things. And there's a critical transition from glycolysis to TCA cycle, basically bridging the pyruvate and the acetyl CoA, and that is done by the pyruvate dehydrogenase complex, which have this mechanism like 5 different steps here. So you should understand each step. And we discussed extensively this TCA cycle, focusing on each step from 1 to 8. So you must understand and need to memorize some of the critical steps and the intermediates here. And this metabolic pathway is basically to generate energy, which is in the form of ATP in the cellular system. And basically we discussed how much of energy or the ATP can be generated in each component of this central metabolic pathway. In part 4 of metabolism, we really focused on the electron transport chain, which can generate a lot of energy, and that's the last step of this central metabolic pathway. And we first talked about the oxidative phosphorylation, which is basically ATP synthesis through the electron transport chain in the mitochondria. In order to understand the mechanism of electron transfer events and the oxidative phosphorylation, we talked about the reduction potential, or the E right here, and its application to the redox reactions. And the standard reduction potential difference in delta E naught of the redox reaction is really important to tell the direction of the such reactions. And Nernst's equation is the modification of this delta E naught to make it more relevant to the cellular system. And this graph shows the E naught prime of the different components of the electron transport chain from complex 1 to complex 4, and also shows how the electrons move. And then we talked about the complex 1, 2, 3, and 4, as well as coenzyme Q and cytochrome C, which are all the critical components of the electron transport chain. So you must know what events are happening there for transferring the electrons within the complex as well as between the complexes. And also, we talked that there are two paths for electrons. One's through the complex 1, initiated by the NADH, and the other is through the complex 2, which was starting with the FADH2. And very importantly, chemoosmotic hypothesis is so important to explain how ATP is generated from the electron transport chain. And then we talked about the ATP synthase, especially its structure as well as the mechanism it generates the ATP from ADP using the proton gradient that was built up by the electron transport chain. And PO ratio, or the phosphate oxygen ratio, is very useful in a way that we can estimate the number of ATP molecules that are generated from the electrons that went through the electron transport chain. And lastly, we talked about the free radicals, which is a sort of a byproduct of the oxidative metabolism, and also talked about some enzymes such as SOD, the superoxide dismutase, that can reduce such toxic free radicals. So for this metabolic pathway, you should have a big picture where you know the inputs and outputs and their quantities, as well as their types of coenzymes and so on. And also, you need to know the details about the the enzymes, their mechanisms for processing the intermediates and so on of each components.